admit additional people as they kind of pop into our lobby area. So hello, I'm Amy Sterling, a crowdsourcing manager here for Flywire. And we are excited for you to join us today for our last workshop of this year, but also to kind of get a tour of some of the new features that have been released in Codex since our last workshop, and hopefully answer some questions and maybe even do a little brainstorm at the end about new features that could be worked into the Codex development pipeline in 2024. All right, so just going to start out with a quick little intro of Flywire. So you're probably familiar with Flywire if you're here, but just as a really quick recap. So Flywire is the female adult fly brain. It's been in the works for a long time. So it was first imaged by Jihao and Davi back in 2018 in Cell. And then our lab realigned and resegmented it. And then over the past 4 years, hundreds of people, hundreds of researchers and citizen scientists and professional tracers have been proofreading this data set. So something around 3 million edits to the automated segmentation has produced a whole brain wiring diagram. So this diagram contains about 140,000 neurons. Newly released since the original preprint came out is the optic lobes have been proofread, and one of the optic lobes has been fully tagged and annotated. And all of the neurons that are in the volume have been assigned to, at the very least, one superclass. There's, I think, 11 superclasses in the volume. We're cycling through them here, and the superclasses, then have their own classes and subclasses and cell types and cell subtypes. So there's an ongoing annotation endeavor from the Flywire community to tag all these cells in the brain, which I think will be going on for some time. And it was announced at SFN from Mala Murthy, but we will be embarking on the brain and nerve cord, the bank data set from Wei-Chung Lee. So we'll have comparative whole brain connectomics in our near future. So that was all cells in the brain. I wanted to highlight just a few, cool sets of cells. You may recognize these cells from the EPG ring. These are some of the auditory neurons, and I wanted to mention these because early in the days of Flywire, Mala published a paper with sort of a subset of these cells, and more recently she's come out with a new paper in Nature. So there's room for iteration and evolution in the analysis of these. These are the CT1 neurons. Axel and Amalia just came out with a great paper, really crazy complicated cells. And then, as I mentioned, we have the neurons of the optic lobe fully proofread. So here's some of the DM4 neurons, and they're tiling the whole eye. And these neurons, of course, are coming with their connectivity graph. So there's something like 50 million synapses, and each one of those has neurotransmitter predictions, which of course can be explored within Codex. And then the brain is further subdivided into 78 neuropil. So these are anatomically defined. Some of them have unique network properties, which you can read about in Albert Lin's paper. But you can use these as parameters for your exploration in Codex. And I think there's a lot of opportunity for discovery at this sort of mesoscale. And then lastly, I mentioned that we have sort of ongoing annotations. There are over 1 million annotations in Codex, so some from our labs, Sung and Murthy labs at Princeton. Greg Jeffries' group at Cambridge added something like 700,000 annotations, including hemibrain matches and hemilineages, and then Flywire community members will hopefully be evolving this ontology and a set of annotations in years to come to give us a bigger picture of this whole brain. So with that, that's a sort of brief intro to Flywire, what it contains, and what types of data can be explored in Codex. So with that, I'm going to pass it off to Ari, who is the mastermind behind Codex, to do a demo of this Connectome Data Explorer and show some of the hot new features that will hopefully aid your exploration into this little treasure trove of data.
Hi, everyone. My name is Ari. I'm a research scientist here at the Princeton Neuroscience Institute, and I work with both Mala Murthy and Sebastian Song labs on multiple different projects. And today we will mostly cover the new functionality in Codex. We have some features that were developed in response to requests from this group and other users. So I will cover them. Then we have a set of questions that we received through the Google Doc that Amy shared. Plus any live questions, if there are, we can answer them in the after the demo. If there's any question about the demo itself, things that are not clear, please feel free to ask, and we will answer them live. Otherwise, we'll refer to all the other questions in the end. So I will share my screen. Yeah, so just a reminder, Codex is a web tool, and the primary kind of reason to Codex's existence is to give access to connectomic data sets and to make access as easy as possible without any requirements or no deep knowledge of the resource or the tech behind the data set. And so it is a little bit different from other similar tools, but the primary differentiator that we're trying to basically design Codex around it is the simplicity of use. At the moment, the data set in Codex is the Flywire central brain. We have multiple versions of this data set loaded, but in future we are planning to have additional data sets available through Codex as well. I will not cover the basic kind of functionality of Codex. There are a few demo videos that cover the basic functionality. So I will jump into what's new since the last workshop, which was maybe 6 months ago. So first and foremost, we have made a lot of progress on annotating the data set. There is a lot more labels for individual cells. So now we have about 70% of the cells in the data set have some identification label. There is way more cell types assigned to cells. These are the curated cell types that we assign internally. Most of the new cell types came from the OpticLobe project, where we typed all the cells in one of the optic lobes in the Flywire dataset. But we also have the ongoing project for typing the double-sex fruitless cells, and these are also pulled into cell types here. So you can see now approaching half of the cells having typed in the entire dataset. Then we have this new connectivity tags. This is also from one of the papers in the Flywire package. This is the paper by Albert and Tony. Cells have these tags assigned to them. Basically, these tags tell the connectivity pattern of the cell, whether they have reciprocal connections or they are form 3-cycle and so on. Then we also have annotations for gene expressions. So this set will grow as well. This is a work in progress. And overall, this page is new. It allows you to explore all of these different annotations that are in the data set from top down, and then you can dive deeper. Say if you want to understand the different cell types, you would go here and click on this and see all the different cell types we have in the data set. And from here, all these links are live. So from here, you can jump over to the cells of this type and so on. So this I already mentioned, the cells in the right optic lobe have been completely typed or almost completely typed. This is a work we recently published with CJ and the and many others in the consortium. And here we have a dedicated page for the catalog of types in the optic globe. You can explore all the different types of cells in the optic globe, and from here you see these nicely made illustrations for how the typical cell looks like and which neuropeels it goes through. You can see some statistics about the cell type, neurotransmitter types, the main regions of inputs and outputs, and so on. And you can also, from here, you have links to jump and see all these cells in the neuroglancer. These are the C2 cells. Or you can see them as a list in Codex and carry out your research from here. Another new big feature in Codex is the ability to search for motifs. This was launched, I think, in June, and it allows to look for substructures in the data set. So maybe instead of explaining in detail what it does, I will just show an example. Say we wanted to find a 3-cycle, basically 3 cells where A goes to B, B goes to C, and C goes back to A, where one of them is a clock neuron. Then we would just type in, say, for the first node, we would say the search query is clock. The other 2 nodes, we would keep them empty. And then I would ask for A to B to form an edge, to form a connection. And then from B to C, as well as C to A. So this way I'm forming a cycle, A to B, B to C, and C back to A. I can enter another set of constraints here, but maybe let me just hit search and see what the results look like. So here you can see there are 3 different motifs that were found, where the A cell is always the clock cell, and then it has connection to some other cell B, and then other cell C, and then back to itself. So this is this 3-cycle is one motif that we found using this tool. And you can also see the motif here in the neuroglancer. There are 3 different cells, and you can see the second match and the third match, and so on. So now hopefully this is clear what the tool is for. Let's go back to the form here and see what are the different constraints we can punch in. So I've only used the constraint for node A, but I can also add the constraints for nodes B and C if I want a certain specific side of the brain or a specific cell class or cell type. Then for the individual connections, I can specify the region. If I'm only interested in connection in a specific region of the brain, I can set the threshold for synapse count, and I can also specify neurotransmitter type. Right, so this tool allows you to basically search all motifs up to size 3 within the dataset. I also want to mention this tool was primarily developed by Ryan. I don't know if Ryan is online, but if Ryan, if you're online, feel free to chime in and add more. Then the next new feature that I wanted to show here, this was requested by multiple Codex users. We sent out a survey a couple months ago asking for feedback on what's missing in Codex, and this was one of them, is the ability to map root IDs of cells from different versions to the current version. So just a quick recap for those who don't know how this data set is built. We have an ongoing proofreading process where we take these 3D reconstructions of cells, and we either merge 2 reconstructions into one cell or split branches into taking one branch from one cell to another cell. And this process is mostly done by experts who are reviewing the shapes of the cells, and they know the morphology, they know how to identify mistakes done by AI or by previous proofreading edits. And so the cell IDs may change from one version of dataset to another version of dataset. If a proofreading touched a certain cell, then it will get a new ID in the new dataset. And so this way we make sure that ID of a cell is also tied to the exact morphology at the timestamp where this ID was issued. And so in many cases, especially in the early phase of the proofreading, we would have these IDs change a lot for the same cells. So even if there was a slight branch removed from one giant cell, we would assign the new ID. And it was a little bit frustrating because you had to kind of manually map these IDs from one version to a new version whenever a new version came out. Today, this still happens because proofreading is still an ongoing process, although it's at a lower rate. And so users were asking for this ability to take any root ID, whether it's from the older version or from a newer version that is not in the snapshot, and map it to the existing version. And so we have implemented this feature here. So say this is a root ID from version 7.8.3, current static version loaded in Codex 630. So we can, if we search for this root ID, then this is the screen we get here, that the root ID is not included in the dataset, but it will suggest us to map it to the current dataset. And now when I click OK here, what it does, it actually goes to Cape client and issues the request to map this root ID to version 630. And in this case, the mapping was successful. The mapping is done based on the closest cell in terms of the components that are contained in the cell. And so from here, this is I think it's a CT1 cell that was mapped to this one. So we can just click and see that this is actually what we were looking for. This should load momentarily, yeah. You can provide a set of lists here, list of cell IDs in this window, and it will map all of them to the current version. Then let's move on to the next thing I wanted to demo. There is improvements or extension to the query language, to the search query language. This is the same query language used in all the different applets in Codex. One of the requests that was also shared in the document that Amy circulated for this workshop was the ability to look for downstream and upstream partners for groups of cells, not just individual cells. And so this feature was recently added. So now you can search for downstream cells of all the MI1 cells. And you can also search for upstream of all the, say, T4A cells. And when you look for upstream and downstream of a group of cells, there is two options. You can either require upstream to only contain anything that's in the intersection, or you can require, or you can ask for downstream from any of the specified cells. So in this case, I'm asking for cells that are downstream of MI1, of any MI1, and the cells that are upstream of any T4A, and search here gives me these results. If you want to say find cells that are upstream of all the specified cells, then you would use downstream intersection, and so on. All this is, by the way, documented here on the FAQ page. If you scroll back to the advanced search section, all these operators have a definition here. So you can experiment with them and see how they work. Another question that was asked in the document was the ability to search by region projections. And so I added this here as well as an example for the query language. In this case, I am looking for cells that have inputs in the lobular plate on the left side and outputs on the lobular plate on the right side, and also adding another constraint that I'm not interested in acetylcholine expressing cells. So this would be this is how the query would be performed. So indeed, you see cells that take from lobular plate on the left to lobular plate on the right. I think this covers also another question that was about looking for neurotransmitter, how to query by neurotransmitters.
And we can send a follow-up email after this workshop that has links to all these queries, just so that you can explore and like play around with them on your own.
Mm-hmm. Yeah, maybe we should just also, are we going to post it on YouTube?
Yeah.
So we can send a link to this video as well. For, then this can be further extended. So I want to just spend a couple more minutes on this advanced query. So for instance, if I want to take this set of cells and also require side is right, the side of the soma in this case, and I will just add this as another term to the search criteria, and I get 4 results. And these logical operations can be applied either in AND, which is conjunction, or as a disjunction of the conditions. Again, all this is documented, and it's a little bit more advanced. It's not for the beginner users, but if you are really looking for some specific set of cells, these allow you to kind of slice and dice the data set easily based on connectivity. Another, and I think this is the last on my list, feature I wanted to share here. This is a set of features basically that we developed while tapping the optic lobe, and it was extremely useful. So it now became available to anyone. So say you have a cell and you're looking for similar cells. Originally, we had these NBLAST scores imported from Philip's work that allowed you to look for cells by similarity in shape. And so, for instance, here I could click this link and see the cells with similar shape, and indeed they all look the same. But oftentimes, and this is indeed what was most helpful in typing the cells in the optic lobe, what you're looking for is not similarity by shape, but similarity by connectivity. And the reason is that if you have types that are numerous that contain multiple cells in different regions of the brain, in most cases, they will not have a similar shape, and this NBLAST method will fail. And so now you have the ability to find cells with similar connectivity. So this is kind of the default and now sits under the NBLAST link. And this will show cells, you see here, these are not the same shape. They will not come up in NBLAST search, but they come up here because they have similar connectivity, meaning that they have similar upstream and downstream partners. And then if you want, again, fine control, more advanced, then you have a more detailed, a more kind of granular ways to find the similar cells. You can find cells that have similar partners upstream or downstream only, while the default is both upstream and downstream. And then another one is where you find cells that have similar input and output types, not just individual cells. And this was extremely useful, again, for finding cells across the brain that have similar connectivity patterns. Then here again, let's try this one. So again, now I'm looking for cells with similar input and output types, not just individual inputs. And indeed, it made the jump over to the other side of the brain because these two cells have the same upstream, downstream types, but not the same upstream, downstream individuals. All right, so I think I will pause here and see if there are any questions about the demo. Let's see.
Oh, that was just me saying, please ask questions.
Okay. All right, so I don't know if people can just ask in audio.
Oh, we had a question. I'm wondering, how do I jump from Codex to the original EM data to look at the synapses?
Right, so let's maybe do this. So for any cell, you can jump over to the Flywire editor, and that would be the quickest, I guess. And then here you can view the EM as well. So again, this is a link from the cell detail page. It has open in Flywire editor. I hope this answers the question. I think
Is there currently a way to see, I don't think that there is a way to see the actual connection points between, say, a cell and its, say, for example, like upstream partners.
no, it's currently, I don't think it's possible in Flywire as well, in the editor. But I
Yeah.
need to double check. But
Actually, Jay fixed the connectivity app, so we are able
It's
to
working.
see. Yeah, so we are able to, if you go to the light bulb menu, and there is something called connectivity analysis, and there's connectivity and synapses with partner. So then you will be able to see synapses from the segment that you select.
so you mean you see synapses annotated on the 3D?
Yes.
Scale? Okay.
Yes.
So maybe
Yes.
CJ, you can maybe after this you can take over and show it on the screen. Maybe you can share it.
Sure.
Yeah.
Sure.
All right, so let's see, maybe one by one. Is there a metric score for similarity in connectivity or shape? Would it be possible to show the score on the threads? Yeah, so indeed, these scores are shown here. So if you look for a similar connectivity, it shows the score. So we use Jacquard similarity as a metric. For every different similarity measure we have implemented, there's a different kind of score associated and this different method. Here we use Jacquard, so you see the similarity here. Same would be with, if you look for, say, similar shape, then the default metric is n plus for similar, and you see the scores here. And they're sorted by this score. So this is Farzan. I hope this answers your question. Okay, great. And then could you also look for cells with overlapping projections? So yeah, this is indeed what I showed before. So so yeah, I hope I understand the question correctly. But anyways, if this is the right question, if this is the question, and if not, please, is audio enabled, Amy?
Sorry?
Is audio enabled for everyone?
Yes, anyone can. You can unmute yourself if you.
Yeah, feel free to unmute yourself if this is not the question you asked. So here I can do, say, upstream intersection of these two cells. So I will be looking for projections upstream that are shared between these two cells. And these are all these cells. They are both upstream of both this one and this one. If this is not the question you asked, please unmute yourself and explain. Right, so about the transcriptome data, I don't think it's on the roadmap for the near future. If I don't know, Marissa, Amy, I don't know, is anything planned, or CJ, anything planned here on Flywire?
I'm not sure.
Okay, so maybe the conclusion is it's not planned in the near future, but if it's again, as with any other feature that is requested by multiple users, you can prioritize and.
Yeah, Sergio, if you have an example of how you would imagine that working, or maybe of use cases of how that would fit into your pipeline, that's helpful in sort of feature development and working things into the pipeline. We do have the genetic lines tool. But,
okay, so I think we covered the questions on chat. If there are any other questions, please drop them here. If not, CJ, do you want to show the how we can visualize synapses in Flywire?
Yeah, I just realized. Yeah, sure. Okay, I just tested. Looks like the EM is not showing, but definitely you are able to see the synapses. So the way you get to it is, let's say you are interested in the synapses from this neuron, and the place you go is go to this light bulb. And I think there are two places you could query synapses, and this one is from a single cell. Then here you will have the ID that you don't need to type in, but you are able to adjust. So for all the annotated synapses has a clef score. So those synapses are from Buman detection. Then individual synapse would have a clef score. You are able to adjust the clef score over here, but usually we would suggest people to use synapses that has over 50 clef score, so you get more of the true synapses in your query. And then from here, you are able to select any of the upstream partner or the downstream partner that you want. Say, I want to see my neuron synapsing with this cell and this cell. And then here you click generate neural glandular link using the selected partner. Okay, I was trying to figure out why the EM is not showing correctly, but here you will see the annotation. Wait, is it
still
right?
Yeah, this
offset
was the
because there's a
same bug.
difference between the
Yeah.
coordinates?
Oh no, it was
It's a cosmetic
working
error
a few
still.
days ago.
Oh no.
Yeah.
I was so hopeful.
Wait. Yeah, it's definitely working. I saw Jay doing it. Okay. Let me see. Let me try another one. Okay, synapse with partner. Okay, so for this one, I will have to say I want to see whether there is any synapse in between this two neurons.
I think this app was developed before Codex even existed. Is that right?
Yeah, that's true.
It's Codex.
Okay, there is no synapse in between them. But A to B synapse.
I think at this point, a few folks asked about this, so maybe we should.
Yeah, so
Codex.
fixing the scale is quick. So I would ask Jay, or we will dive in why it's not working again. But here would be the place that you going for synapses. One is from here, and here you could look for synapses with partners. And we will definitely notify you guys when it's fixed.
Mhm.
Yeah, sorry about that.
Thanks so much. This is really helpful. We'll play with it and see our neural interest. Thank you.
There was another question from
Olivia here.
I will go back to sharing the screen.
Is there a way to view downstream or upstream commonalities between multiple cell types with one search?
Right. So this is there's multiple ways to do it. So say we want a cell type is say LC14 or I don't know cell. What else is a good cell type? So this is just a query to grab all the cells that have either this or this type. So this is just a step one. Now we found 446 cells of this sort. Then we can jump over to stats. This will take us to statistics about this query. And then here you can see top output types. These are the output cell types with the most synapses to this set. And you see the top output type is this one, and then the M3P, then M3P, and so on. And there is a top input types as well. These are upstream types. So this would be the easiest way to achieve this. In general, anything that you can search for, you can also do all these different operations for. So you can view 3D skeletons of all the cells matching the result. You can do a connectivity network of the cells. This will take you back to a list and so on. If the results are not too many, say up to dozens, then you can also see pathways, and these are more intensive operations. Hope this answered the question. Okay. Yeah, I don't think there's any other question here. Did I miss anything, Amy?
I don't think so. Just Farzan showed a link that worked, so you guys can check that out to see the synapse annotations in 3D.
Oh, great.
There you go.
So what is the input to this? It's a cell ID?
Not sure. Actually, that was from Farzan.
Okay. Anyways, yeah. So we will probably need to look into it and see if we can easily implement it in Codex. That will be convenient, I think.
Well, that might be a nice little segue to well, while everyone's here, if anyone we are always looking for, feature suggestions, and also user feedback on things that are things that are frustrating while you're using Codex or Flywire. I'm sorry, my cat just let out a crazy yowl. I'm sorry, I lost my train of thought there. But while everyone's here, does anyone have, as you've been using Codex and exploring this connectome, are there any features that you're just yearning for that would really help you in, exploring these data and asking questions? Like what are some things that we're missing that would be beneficial to the community? Feel free to unmute yourself if you have any ideas. You can also post things in chat. Like the one example of seeing synapses was sent in a couple of times. Well, if we have no feature requests, you guys can sit on it and think about what we could do to make this tool better. And feel free to email us at any time. I guess, this is essentially going to conclude our workshop for today, showing these new features and doing Q&A. But I just kind of wanted to close by saying thank you for using these tools. And they really rely on your input and your suggestions. So feel free to email us, Flywire at Princeton, or Slack us, or send us your feature requests and questions, and we're happy to address them, and hope to build more powerful features that will make this resource more valuable to everybody in the future. So with that, we'll wrap it up. I hope everyone has a nice beginning of the holiday season. And we'll see you guys hopefully at the next town hall that'll take place sometime in January. So thank you. Bye-bye.
Thank you, Ron. Bye-bye. See you.
Amy,
Yeah.
one of the audience asked whether we will have recording.
Yes, this is
Oh.
being recorded, and we'll upload it onto the Flywire YouTube channel, and we'll email it out to everybody who attended today. So speaking of that, let me stop the recording.